So I'll be talking about my project titled Characterizing Landscapes of Somatic Mutability using a graph-based approach. Okay. So to briefly introduce the problem, we know that the human body is an evolutionary mosaic. And what that means is when cells are dividing throughout your lifespan, they're undergoing an evolutionary process, just like any other evolutionary process on Earth. And what this does is it creates clones of different kinds of cells, which ends up creating this sort of mosaic, or you could conceptually think of it as a mosaic of the human body, which I pictured on the left there. And the most important ramification of this is cancer, which is a disease of somatic evolution. So basically, healthy cells in your body will accrue mutations over time, and some of those mutations will give cells selective advantages. And over time, through selection forces, eventually those cells will grow into tumors, and later, through further evolution, they can grow into metastases and eventually kill you. So in understanding somatic evolution, we have to think about what drives that process of evolution. And the most obvious one would be mutations. But there are many different kinds of mutations that can occur through clonal evolution. So there's the more standard single nucleotide variation, which is just a change in a base of DNA. But then there's also more complex forms of variation, such as copy number variation, where you can have multiple copies of a segment of DNA or gene. And then structural variation, which is where you can have rearrangements of the genome that produce very novel forms of variation and possibly higher selective effects. And then also epigenetic variation, which is less understood, but is gaining more traction, which can be things like chromatin conformation or methylation and things like that. So in understanding the evolutionary process, you have to also think about the evolutionary dynamics of these populations. So in terms of evolutionary patterns, you can have different forms of branching, such as linear branching or punctuated branching. And the way that these evolutionary patterns occur is going to shape how the overall clonal mosaic is going to look at the end, which is shown in this picture. And then we can also think about clonal distributions and how they're changing over time. So for example, maybe a particular cell with a mutation is very infrequent early in cancer, but then because it's selectively advantageous, it's growing over time. And then we can also think about spatial dynamics, which is important in development. So you can basically think about how cells are propagating throughout the tissue and forming these sort of geometric patterns. And then finally, the last form of evolutionary processes I want to talk about is mutability rates and patterns. So this is becoming a hot area in cancer research, but essentially the DNA recombination repairs and endogenous and exogenous forces from those create these sorts of patterns on the genome. So you can see that this sort of distribution over mutations and locations and mutation rates end up changing throughout the lifespan and due to different forces. So you're essentially creating a form of evolution which has downstream ramifications. So one point I want to emphasize in our study of somatic evolution is that it's a very challenging problem to trace the evolution of a cell population throughout the body, as you can imagine, because the cell patterns are present at very small frequencies. You have to collect tissue samples and sequence those samples, and there's no uniform way to do that at high coverage or through examining each single cell. And then on the computational end, there's a lot of different algorithms that go into calling variants, calling structural variants, and none of these are at all solved problems. And the amount of data is a tremendous challenge in examining somatic evolution. So when we're thinking about how to analyze it, most of us are familiar with the sort of standard genomics analysis framework you could think about in terms of a GWAS study. So you take a bunch of variants, you align it to the genome, and then you do a statistical cohort analysis. You'd say, these people have this variant and they have this disease condition, these people don't. Then this variant is implicated in the disease and so forth. But I want to emphasize that this sort of analysis is very likely faulty in the case of somatic variation because of the fact that we can't really properly align a lot of these structural variants. And basically call these mutational patterns in order because for one thing, they're very low frequency, and for another, the sorts of variation that you find in cancers can be extremely dramatic, like a whole chromosome rearrangement or something that you just wouldn't find in traditional genome aligner. And then there's also just different sorts of information that we have to find out in terms of evolutionary patterns, which are more important in the cancer case than in the individual case. So we care more about things like phylogenetics or mutational processes, which are driving the evolution of cancer, than we would say maybe if you wanted to analyze a newborn baby for Down syndrome or something, you wouldn't care about that. But so this is just to emphasize there's a tremendous difference when thinking about inter-individual variation. So the difference in variation between me and someone else versus intra-individual variation, which is the population structure of cells within an individual. So I'm just going to focus on the somatic variation portion, but we care that the fact that variant frequencies are often changing over time. We care more about the evolutionary structure and the mutational patterns, and we care perhaps less about the individual variants themselves, since they can be extremely hard to call. Yeah. So the basic premise behind this preliminary work is how do we quickly characterize the somatic evolutionary landscape of a tissue without necessarily, let's say, performing a disastrously huge computational analysis of every single cell in the body or something like that, right? So the basic idea behind this is that if we create a genome graph, then perhaps the structure and the properties and statistics of the graph can let us can give us information about the sample without going into very specific forms of cellular variation. So we wouldn't need to call, let's say, like a particular variant in a particular cell. Instead, we can just look at it in a population sense and deduce things about the mutational patterns or the structure of the cell population without doing that sort of analysis. So just briefly laying it out further, we take a set of reads and then we simulate a somatic evolution of cells, which basically gives us a ground truth for how or what exactly the evolution is going on. And then we build a k-mer graph by essentially taking the set of k-mers and creating an edge if the k-mers differ by one base. So that's just showing you if they're connected in the actual genome. And then we weight the edges by the frequency of each connection. So that's important because, like I said, the cell clonal frequencies are very heterogeneous. So you can have variants present at very low frequency, which are very important. And then we basically count these sort of subgraph cycles and motifs, or which we label by the frequency along each edge. And we're going to do this, but using these derived statistics, we can then say things about perhaps how the cell population is evolving, maybe if it's cancerous or precancerous or things like that. So these are just some examples of how the graphs look like. So the graph on the left is, these are obviously miniaturized because you can't see it on the whole genome, but the graph on the left would just be a very low frequency somatic single nucleotide variant. So there's this big bubble and this little bubble here, which are two single nucleotide variants which occurred at low frequency. And then the graph on the right is chromothripsis, which is this large-scale shattering event. And you can see how there's a lot more variability in these bubbles or things like that. And so a key part of this analysis is the fact that these subgraphs are giving us information about the kinds of variation. So there's this form of variation called contagious, where you have hypermutability of single nucleotide variants in a very short region. So it looked like this in the graph. But then maybe if you have a big inversion, it's going to look more like this large oval segment combined by the two ends. And then we can compute statistics, which are giving us information about the kinds of variants, their frequency, and basically where they are in the genome. I'm not going to go through all of these, but things like degree distribution can be useful, cycle lengths, the weights, their density, things like that. And the basic idea behind this analysis is that we'd be able to use the graphs in this sort of walk through the somatic evolution landscape. So if you're thinking about the start as a healthy cell, then maybe you start with this graph on the top right. And then as you progress into the cancerous stage, the frequencies of variation are changing, and you'll get this sort of graph structure on the bottom right. And this is a hypothetical drawing, but it illustrates the kind of point there. And so just to wrap up the presentation, We believe that these read graphs and statistics and motifs may give us a quick way to view the properties of somatic evolution landscapes, and future work is going to involve benchmarking this on real data sets and potentially exploring the further graph properties and statistics that might be useful in tying it to real somatic evolution properties. And with that, I'd like to thank my advisor, Russell Schwartz, and all the members of the Schwartz Lab, as well as the funding sources. Thanks.